Hey everyone, in this lecture we are going to learn about the myocardial action potential, and this is going to be very important because this will help you to understand the first and foremost important thing, that how does the electrical activity happens out in our heart, right? And how does the different phases they do help out in the generation of that action potential, right? And most important clinical term, the clinical concept that we are going to actually look up to, that is, why does the cardiac muscle cell doesn't go the tetany, right, just like the skeletal muscle cells, right? So let's just look up to the basic idea before we start understanding all of this graphical representation of this concept. So do remember that every cardiac muscle cell is surrounded by a cell membrane, definitely separating the inside of the cell from the outside, right? And across this membrane we do have one of the electrical charge. So this difference in charge is called as the membrane potential, as we do now, right? So at the rest, the inside of the cardiac muscle cell is more negative compared with the outside. At rest, the inside the inside of the cardiac muscle cell is more negative compared with the outside, right? And that is why the cell is to be called as polarized, right? Yeah, and the membrane potential changes when the ions move across the membrane to the ion channels, right? So in this topic, we are going to touch up the three important ions, that is the sodium ion, the potassium ions, and also the calcium ions, right? And these ion channels, they do actually move through, these ion channels, they move through the specific, these ions move through the specific channels, and their movement actually creates the action potential. So there are a few terms which are important to understand. Now, the first one, that is the depolarization, right? It means that the membrane is becoming less negative, or it is moving toward to become the more positive, right? Then we do have the repolarization. So this means that the membrane becomes negative again, right? Or it is getting more and more negative, right, after the depolarization. And also we do have the plateau stage, right, which is also being mentioned over here. This plateau stage means that the membrane potential remains elevated for a small time, right, instead of falling immediately. And that you will look up and observe in the phase 2 of this cardiac action potential. All right, so these are the important terms to understand the overall graph. Okay, so now let's first begin off with the resting membrane potential. That is actually the resting state of the cardiac muscle cell. What is the resting state of cardiac muscle cell? The resting membrane potential of a working myocardial cell. For the working myocardial cell is about minus 85 to 90 millivolts, right? That is the normal resting state, right? At this stage, we can say that the cell is not inactive in the sense of being useless, but it is actually in a ready state and prepared to respond to a stimulus, any sort of a stimulus. So this resting membrane potential is mainly due to the high permeability of the membrane to the potassium ion. So at rest, potassium has a major role, right, in maintaining the negative membrane potential, and this resting state later corresponds to the phase number 4 also, right, of the action potential. So first, briefly, for those who are very much new to action potential, they have to understand that this is one of the rapid and temporary change in the membrane potential caused by the movement of the ions across the cell membrane. And it is very important to understand that this graph is actually representing the electrical activity of the single cardiac muscle cell, right? So do also remember that this is not ECG, okay? This is not actually the ECG, right? And this is just the action potential mechanism. And one of the important distinction is to be making out over here clear that action potential is the electrical event in the one single cell, but the ECG is the recording of the summed electrical activity of the heart from the body surface. Okay, many students actually they do mix up these two, so you this point must have to be very clear. And now we have to understand that where does the myocardial action potential, where does the myocardial action potential occur, right? And first thing that you have to remember it out, that it occurs in the atrial muscle fibers, right? It occurs into the ventricular muscle fibers, right? And Purkinje fibers, right? It also occurs in the Purkinje fibers. And do remember that the myocardial action potential does not occur in the SA nodes and the AV nodes, okay? doesn't occur in both of them. This is very important because the nodal cells, they have a different kind of action potential, and we will just learn at the beginning specifically the working of the myocardial cells in the ventricular myocytes. Okay, so let's look up to the overall phases. We do have the phase 0, 1, 2, 3, 4 that you're looking up over here, right? And these phases, they do represent the sequence of the voltage changes occurring across the membrane. So now let us study one by one. Okay, the phase number 0 that you're looking up over here, this phase, you can remember it as the phase of the rapid depolarization. This phase is for the rapid depolarization. This is the real phase where membrane potential actually rises very sharp from minus 85 millivolts, right? And you see that rising very much sharply to the phase number 0, and we can just simply say that this is the rapid depolarization state. And the reason is that the fast voltage gated sodium channels, right, the fast voltage gated sodium channels, they do open quickly, and that is why the rapid stroke and the depolarization is being seen from minus 85 to the positive one, right? And these the sodium channels open, the definitely sodium channels rushes into the cell. Since the sodium is a positively charged ion, its rapid entry makes inside of the cell much less negative and then positive, right? And this is why the graph shows a steep upward stroke that you are actually looking out and observing here. And another very clear logic is that the sodium channels in the phase 0 opens, right? The sodium enters rapidly and the positive charge enters. So the membrane potential rises sharply. That is the phase 0, okay? Means that this is actually produced. We can just simply say that the phase 0 is being produced by opening of the sodium channels and bringing more and more sodium influx. Right, so this phase is important because it determines the rapid electrical activation of a myocyte. Later on, we do have the phase 1. What is the phase 1 to be? Initial repolarization. It means that to get it back to the real state, right? And after a riser into the upward stroke, right? Initiation of the myocardial action potential in a myocyte. What happens? That the membrane potential shows a small downward notch. Why is that? Because you see that once this green one that you're looking up to the upward phase, right? This is all the upward phase because there's a rapid upstroke and depolarization. What happens in the phase 1? We actually observe very small, you can look up over here, a very small downward notch, right, in which is this yellowish one that you're looking up over here. Now why is that is happening? Because the fast sodium channel starts to get inactivated. So inactivation of the voltage gated sodium channels happens out, right? And the rapid inward sodium current stops, right? So the influx stops and the curve goes a little bit downward. At the same time, there is a brief outward movement of the potassium too. So now sodium entry decreases, right? And the potassium starts leaving the cell. So the positive charge is moving out. Of course, the positive charge is moving out of the cell. And what is happening? The positive charge is start moving out and the membrane is getting potentially falling slightly, right? And that is why the graph is showing up a small dip, right, after the spike. So the phase 1 is actually a short transitional phase, right? And the key idea is of all this phase that the phase 1 is due to the brief, right, to the brief potassium efflux, right? And what is going to happen now? There's a small dip that you're looking up over here in this yellowish format. The phase 2 represents the plateau phase, okay? And what happens in the plateau phase? This is going to be the most characteristic part of the myocardial action potential, right? And this phase makes the cardiac muscle action potential very different from the skeletal muscle action potential. And in this phase, what is going to happen now? The L type, we do have the long lasting, right? The L type calcium channels to be open up, right? We do have the L type calcium channels, and these channels get open, right? And what is going to happen out? The calcium is going to enter the cell. When the calcium Ca2 plus is entering the cell, During this same time, potassium is also leaving the cell. Potassium is also leaving the cell. Now, this is the point that if only potassium were leaving, the membrane would rapidly repolarize, right, quickly. But if only calcium were entering, the membrane would remain strongly depolarized. But here, both events are happening together. The inward calcium current and the outward potassium current. So these two current balance each other for some time, and because of this balance, the membrane potential remains almost flat, remains almost a little bit flattened, right, for a small time, right. And this flattened, or you can just say that this membrane potential remains almost flat instead of immediately falling, and to be known as that we do have a plateau phase over here, right? Why is that? Because the two events are occurring at the both same time. That's why you can look up over here for the calcium and the potassium itself, right? The inward calcium current and the outward potassium current, right? So it means that we can just say that in the plateau phase, what actually happening out, we do have the Ca2 plus inward flux, inward, right, and the outward of the potassium, right, making out the membrane to be a little bit in a flattened phase that you're looking up over here in the blue color, right. And do remember it out that whenever we do have the channels which bring up, for example, the L-type calcium channels open up, right, you have to remember that the sarcoplasmic reticulum actually stores our calcium. And in our previous lectures, we have also discussed about the excitation and the contraction coupling that's been actually happen out usually whenever we do have the calcium influx, so that the cardiac muscle cells can also get the contraction. Now, when we do have the phase 2 to be get activated, and we do have the plateau phase, we have to understand that why does this phase 2 is to be considered as very, very important. Why is that? Because this is going to be having one of the functional significance. The calcium that enters the cell during the phase 2 does something crucial. It triggers the release of more and more it triggers the release of more calcium from the sarcoplasmic reticulum, definitely, right. And whenever we do have the more calcium from the sarcoplasmic reticulum, all this is actually known to be as the calcium induced calcium release, right? Because in the beginning there is less calcium being coming inside. Those ones are not enough to actually generate the action potential or depolarization, but the smaller calcium influx that triggers the more calcium release, right, known to be as the calcium induced calcium release, right. So the sequence is that the calcium enters from the extracellular fluid. This triggers more calcium release from the sarcoplasmic reticulum. The intracellular calcium increases. The calcium binds to the troponin. The actin myosin interact. The contraction happens out, and all of that is actually known to be as the excitation contraction coupling. We have discussed this all in very much details in our previous lectures, right. After that, we do have one of the important phase, that is the repolarization state, right? And that is the rapid repolarization state, the phase number 3. What is happening over here, that after the plateaus, the membrane potential starts falling back toward the resting side. You see that starts falling down. It is going to back to the resting state. And during this phase, the calcium channels, they begins to close. The calcium channels begins to close. That's why the closure of the voltage gated calcium channels happens out over here. And what is going to happen out? Inward calcium current decreases, right? Now the calcium current is decreasing, but still the potassium is moving out of the cell. So now there is no longer enough inward positive current to balance the outward potassium current, right? So potassium efflux becomes dominant as positive charge. As the positive charge is leaving the cell, right, what is going to happen out? The cell membrane becomes the negative again. The cell membrane becomes negative again, right? And that's why the graph is showing you that it's been falling down, right? So the logic of the phase 3 is the calcium entry decreases and the potassium efflux continues. Outward current dominates and the membrane starts repolarization. The phase number 4 that you're looking up over here is to be known as the phase of the, again, resting membrane potential. Means that after the repolarization is complete, the cell returns it to the resting state. And the phase 4 is the stable resting membrane potential of the working myocardial cells. So the membrane potential is again around this, as you can see that. You see the phase number one mean the similar to the minus 85, right? And you can just try to understand that at this stage the potassium is important both in maintaining the resting state and also in the repolarization, right? And this is the different from the pacemaker cells like ethanode, where phase number 4 is unstable and shows the spontaneous depolarization. We will connect that in our upcoming lecture too. So these are the 4 different phases that you have to actually understand. Now we have to answer one of the questions, that why the cardiac muscle cells has a long action potential. Long action potential. Why does the cardiac muscles, they do have the long action potential cells? The answer to that one is that compared to the skeletal muscle, they do have the much more action potential because of this plateau phase. Because of this plateau phase. In the skeletal muscles, do remember that the repolarization occurs quickly after the depolarization. But in the cardiac muscle, the calcium entry during the phase 2 delays this repolarization, and you see that this is the delay reason. So the action potential becomes prolonged, and this prolonged action potential is one of the unique features of the cardiac muscles. Another thing that you should notice is the refractory period. What is this? Now we do have the effective refractory period is to be defined as a period during which the cardiac muscle cell cannot be excited again easily, right? And why is that? Because action potential is prolonged, refractory period is also prolonged. The refractory period is also prolonged because of this plateau, right? And that's why you see that the difference is actually been increased over here with the 200 milliseconds, right? And this is very important for the cardiac function because the heart is a pump. It must contract and relax in a coordinated rhythm. If the cardiac muscle could be repeatedly stimulated very rapidly, it could go into the sustained contraction. And that would be dangerous because the heart muscle relax between the beats to allow filling. So long refractory period protects the heart from the repeated rapid excitation. Somebody can simply ask you, you see that they can just simply pick out whether you have noticed down why does we do have the effective refractory period. Because of this plateau phase, we do get some of the time, if this plateau phase gets prolonged, this effective refractory period gets also prolonged. And this is actually physiologically important because the heart, definitely working as a pumping organ, right, must contract and relax. So for that one, between every beat, the heart muscle actually gets some of the relaxation to fill up. And that is why the long refractory period to be considered as the protection of our heart from the repeated rapid excitations, right? So another one that I've said actually in the beginning, that was about the tetany. That was about the tetany, that why the tetany does not occur in the heart. So you have to remember it out, that what does tetany means? Tetany means sustained contractions. Sustained contractions. What does sustained contractions mean, and how does they occur? The sustained contractions actually do occur by the repeated stimulation before the muscle has time to relax. And this can happen in the skeletal muscle because its action potential is shorter and its refractory period is also short. But in cardiac muscle, because of this plateau phase, makes the action potential a bit longer. A long action potential causes long refractory period, as I've said before. So as a result, one contraction is almost complete before the cell can be stimulated again. So the contractions cannot simulate in this way, and the similar way they do actually in the skeletal muscle format, right? And definitely the tetany cannot occur in the heart, and this is essential for the survival because a tetanized heart would not be able to pump the blood effectively, right? This is important. And now if we just compare the skeletal muscle, right, with the cardiac muscle, we have to understand that in the skeletal muscle the action potential is shorter. No true plateau phase. No true plateau phase, right? And the refractory period is also shorter, right? In skeletal muscle, tetany can occur, right? Can occur. and all of the opposite one happens in the cardiac myocytes or the cardiac muscle cells. For example, the action potential is longer, refractory period is being present, plateau is being present, and tetany does not occur. This is one of the key significant. Another point that you have to remember it out that occurs in all cardiac myocytes except for those in the AC nodes and the AV nodes, as I've said you before. But you have to remember it out that in the working myocardial cells, right? In the working myocardial cells, for example, the phase 0, the phase 4, right? And we do have the plateau. Okay, the phase 0, if I say that, that is due to actually fast sodium influx because of this one. The phase 2, of course, we do have the plateau being formed over here, right? We do have the plateau stage. And the phase 4 is actually stable. But in SA node, right, mainly the phase 0 is due to calcium influx, right? And also the phase 4, this is unstable and this slowly depolarizes its on. All right, and there is no plateau like that of the ventricular myocytes. So in this lecture, that we are only and only talking about working myocardial cells, but not the pacemaker cells. The next lecture is about the SA nodes and the AV node, or you can just say that the pacemaker myocardial action. How does it all actually happens it all? I hope so that this is clear. Any questions you can just ask me during the class, or you can put down the comments below.